something, or I know it's I think let Bonnie introduce our speaker. I just wanted to get my thing out.
All set, people on the internet. Jessica was here. Jessica. So this one only goes to the internet. It doesn't go to the room. I've been told. Yeah, go see if she. Yeah. That was weird. That was like having somebody buy a beer for you or something.
What?
The distance effect. I haven't seen Sarah in a long time. She worked with us in Australia.
I know. Maybe we should. It's going now, or is it? Okay. Well, so it's my pleasure to introduce to you Dr. Robert Dunn, who is our first speaker in this webinar series. And Rob joined us from North Carolina State University today, where he's currently an assistant professor. Rob is a versatile ecologist with broad research interests, which are motivated by a curiosity in understanding and interaction of species and the ecological consequences of these. His focus hereby is mostly, but not exclusively, on ants. In addition to his academic career, he's also a prolific popular science writer and has published numerous articles in, for example, the National Geographic, Natural History magazines. And his second book is due to come out in June this year, with the title of The Wildlife of Our Bodies, Predators, Parasites, and Partners That Shape Who We Are Today. So there would be much more to say about Rob, but I'd rather keep this short and let him tell his story in his seminar on using collaborative approaches to see geography and of life, lessons from ants.
Thank you, Bonnie. I've had a great time already today. I do have to say I arrived at 5:30 this morning. So if I say anything totally inappropriate, it's just the sleep. I decided instead of ants today to focus on the history of children's books. Actually, so this is a children's book that I really loved as a child. And it's a book in which part of what happens in the story, that there's a little boy who comes to the ocean and he meets these brothers, each of which has kind of a non-standard magical talent. And one of the brothers can swallow the entire ocean. And so there's a moment in this story when he swallows the entire ocean, and it's all revealed, everything on the ocean floor. And as a little boy, this part of the story was incredibly fascinating to me. Because here was this moment when you saw all of the living treasures, and maybe a pirate ship or two, here in the bottom of the ocean floor. And it was and remains kind of magical, this part of this story. The sense that you could pull back the veil and see what people weren't seeing. And then if you do it in the right way, that this is possible. And so in part what I want to talk to you about today are ways of pulling this veil back and aspects of the ways in which I try to do this in my career. And part of the way I do this is to think about science in general, to write about science in general, about discovery in particular. And I'm really interested in the process of science and how we make big discoveries, how we as scientists pull the ocean back and see those treasures. And we tend to think of, tools like this big ship, but there are also lots of other tools that allow us to see other things we wouldn't otherwise. And so this is a hand-colored SEM from part of a National Geographic article, and this is done by Martin Ogerle, and he does these beautiful SEMs. And you can see in the coloring aspects of this insect egg that you wouldn't see otherwise. And so even things like art and writing can be tools for seeing. And so in a way, what I've tried to do in my writing is to shine a kind of light, not quite as dramatic as the ocean being brought back, but to shine it on the living world in general. And so I've done lots of writing for general audiences, which I really love every aspect of. And sometimes it's about ants, an article about honeypot ants, but lots of times it's not. And my first book dealt with this broad sense of discovery and pulling back the ocean, pulling back terrestrial realms to see what we can see. More recently, this is the book Bonnie mentioned. I've tried to think about this in the context of our own lives, sort of revealing what lives on us and influences us in ways that we tend to miss. And so if we did the effect of pulling back the sea of our own lives, what sorts of creatures would be revealed? But I've also become conscious in doing this writing that there's still an incredible amount out there to be revealed. And often what we need to do to reveal it is to use some new kind of tool. And one of my favorite examples of this is work that Martina Kalsunas-Rupel at UNC Greensboro in North Carolina has done. And she was working with bats as a young scientist, and so used ultrasound techniques to listen to bats. And then she got interested in mice. And at some point in her career, she thought, I wonder if the mice are communicating with ultrasound. There wasn't any real reason to think this. There were little bits and pieces in the literature that maybe something was going on. There's a 1934 paper that reports on a singing mouse from Detroit. It was caught in a house. It was shown to sing. It was taken to the University of Michigan. A trained musician was shown the mouse. She said it had poor pitch. It was killed. Nothing remarkable was found about it. But the idea that mice might communicate in the ultrasound, there was a sense that it was possible. But nobody had really looked. And so sure enough, Martina went out into the field, has recorded mice, and has shown that Peromyscus mice not only communicate in the ultrasound, but have melodic songs. But the species she's focused on had no fewer than 6 songs. All the other 3 species she's now looked at also have songs. And so it looks as though many, many rodents have songs, like bird songs, and nobody knew it until just a couple of years ago. And so to me, this is really like pulling back the ocean. The idea that all around us in this building, whatever rodents or any around the whole building this is, but maybe there are a couple in here, that they're singing and we're not noticing, and they've been doing it all along and that nobody noticed. I think it's pretty wonderful. And so what I want to talk to you about today are some ways we've been trying to think about the world of smaller things, to sort of use new kinds of tools to pull back to see and see some things. And I'm fascinated by the smaller life. I mean, I even love dust mites. I love how they're always facing you. They never seem to be retreating. They're always advancing. And roaches, and most of these are images by Peter Neskrensky, who's a good friend, and he had the desk next to me in grad school. He's gone on to take pictures, and I've gone on to write, but we keep doing things together. And they're just these marvelous creatures. And And one of the amazing things about the smaller life forms is that we all know in this group that they represent a huge proportion of species on Earth. We get the ants, we get the flies, which I think are particularly poorly appreciated. About 1 in 10 known species on Earth flies. We get the beetles, and this is a little bit of a cheat. Can you see that well enough? Everybody see the beetle? Sure. So can you see the ants? So here's an Aceton burchellii, and then here's the beetle, an ant mimic beetle living with the ants. And there are more kinds of ant mimic beetles than there are bird species on Earth by most estimates, right? So these incredible diverse radiations that I just love. And you know, we often talk about how few species have been named of these radiations of small things. And so you see estimates in the literature of 2 to 3 million unnamed species. You know, the 2 is a pretty conservative estimate. Although if we're thinking about this as sort of a sky equivalent, that would mean that this much of the sky is dark. So even that conservative estimate means we're pretty ignorant. If we get to 30 million, we'll get to be really ignorant. But one of the amazing things about this ignorance is that I think that it matters in a functional consequence for how we live in the world, even if we don't know about it. And so there's some really good examples recently of species that didn't have names. Come on in. Yep. Take a front seat. We all said something embarrassing about ourselves when we came in. That a lot of these processes we think about small things doing are carried out both by species that are named, but maybe predominantly by species that don't have names. And so we have, you know, good ecological processes like pollination, decomposition, pest and biological control. And then we also have these bad processes, these disservices that the small things take a lot of credit for. And these include things like mosquitoes that don't have names that vector diseases. So when West Nile was first turned up on the radar, one of the main vectors of West Nile was not yet named. And so this great big mass of unknown things has consequences. But it's really difficult to study. And I don't do systematics, but anybody who does can tell you that the work of naming these species is incredible. And it's not, there's no fast about it. So how do we think about what remains to be known? And I would say that the part of the difficulty is in seeing when so much has yet to be revealed. And so I want to walk you through 3 ways we've tried to see these small things and consider them. First, seeing through collaboration. Second, seeing through simply looking around. And this seems silly, it's so simple, but I'll argue it's very important. And then seeing with great big experiments. So the first thing I'll talk about is seeing through collaboration in the context of thinking about things like the patterns in the diversity, consequence, and biomass of small things. What they look like even, what their consequences are, and why. You might think in a lot of senses that we know a lot about patterns of things like diversity or biomass, and that would be true for birds or mammals. And what we tend to do is assume that if we know about what the top of this iceberg, if that's the birds and the mammals, that what's at the bottom of the iceberg does the same thing. And that may be the case, but not always. And so part of what we've tried to do is to start with some of these small things that are really consequential, like pathogens, and to see if we can model pathogens and their global distribution. Am I loud enough? Can you guys hear me in the back? See if we can model the global distribution of pathogens and their diversity and consequences, and also vectors, if we know something about the top of this iceberg. So using the top of the iceberg as a kind of tool to see. And what we find is, in fact, if you know the number of birds and mammals in the political region, you can predict the number of human pathogens. You can do a pretty good job. So birders beware. Those places with the most birds also have the most gnarly things that will kill you. But one of the things to notice here is the spatial grain at which we can do this analysis. So we're doing this analysis at the scale of political regions. And so we have the United States, this huge political region, which includes Alaska way up there. And the reason we have this is because the data for pathogen distribution at the global scale, human pathogens, are far worse than the data for, say, warblers. There aren't fine resolution data with the exception of things like malaria, maybe dengue, yellow fever, just a small handful. And so we're really ignorant about these patterns of distribution. And so it's a real problem. I've really been sort of doing battle with to think about how we move forward from here. And one of the models for moving forward is the model that the Breeding Bird Survey has used. How many of you know about the Breeding Bird Survey? Just a quick show of hands. Is this a thing? So this is a survey where citizen scientists go out and survey bird routes. And so each one of these is a route. They listen for and look for birds and document which bird species are in a given place each year. And so it's an amazing data set that results. There's all sorts of biases. People identify birds wrong. People hear some birds more easily than others. And yet it's a really powerful tool for seeing what's going on and change through time. But the problem is, how do we do this for smaller things? The breeding ant survey is not going to work in the same way. Maybe there's a version that could work, but not in quite the same way. And one of the real difficulties is that people don't know a lot of these species anymore. Although I would argue that there was a time when you could have gone around the world and surveyed people about what species lived around them, and they would have been able to tell you a lot more. There was a kind of golden age of local knowledge, when you could have surveyed, for example, indigenous groups and said, well, how many species of ants, how many kinds of honeybees, honey-producing bees do you have? And people could have told you. And so, for example, the Yanomami have traditional knowledge of 32 kinds of ants, or did have, 48 kinds of wasps, and 57 kinds of bees, which roughly coincides with, say, knowing the genera. And so this is an incredible knowledge. And if we actually knew this for every place on Earth, we would know more than we know today. So there was a point when collectively we actually had a breeding ant survey, or just an ant survey. But we don't anymore. And so how do we get around this? And the problem here is this sort of Tower of Babel problem of languages and cultures and knowledge in different fields and different places diverging, and we need some way to reunite them. And so Nate Sanders and I, and now a much larger group, brought together a group of people over beers and said, what if you can tell us about what you know about ants in your place? And we collectively bring together knowledge of the world. And so all these people signed on to this, to sort of use this sort of traditional knowledge of ants, in a way, scientist traditional knowledge, to see what the world looks like. And I'll admit right now, a lot of this is going to be about ants today. I'm biased, but there are some good reasons for studying ants. They're like us, be models of society and disease, and across many cultures, people think of them as like us and resembling us in a way that a lab rat resembles us, but in a social sense. At least where they've been studied, their diversity patterns, for example, predict those of other groups. Maybe they're a useful proxy, maybe not, but beginning. They're ecologically significant. I like them. And ant biologists, for the most part, are more or less likable, which that's a really important part of this, that you can get a bunch of ant biologists together and they're not all mean. So you couldn't do this with some other groups of insects. And you can think of your favorite groups, but there's another social insect group that forages in the soil, and I think you couldn't do this with them. And so we've made some progress here. So these are all data points from individual studies from the global collaborators that we now know the ants present. And they're from different sampling methods, and so we have biases. It's not as great as the Breeding Bird Survey, but it gives us a picture. Just what does the world look like from the perspective of ants? You good? And so we can start to look at really simple things that people have been doing with birds for a long time, like patterns of diversity. And so bigger circles here are just more species. This is ground foraging ants. Smaller blue or greener circles are fewer species. And we get this strong tropical pattern. We can model it, and one of the things we see is that dry places are also really diverse, which is quite different from, say, birds or mammals. We've made some headway in thinking about this. We've also begun to look at canopies, using people's canopy samples to look at canopy ants, and we see similar sorts of things, although precipitation becomes much more important. We have models and maps now of canopy ant diversity. And then we get to this question, and Andrew's actually in the back of the room here, the head in question. Now we get to the question of why we see the patterns we're starting to see, and this is a whole other debate. And I would actually argue that it's much harder to see why these patterns are being generated, and maybe impossible on the broad sense if we're thinking about all groups of organisms, birds and mammals and insects. And I didn't used to think this, but Andrew and I did a kind of survey of people who've worked on large-scale patterns of diversity. And we asked, do you think there's a consensus near at hand about why these patterns emerge what's the consensus? And almost everybody thought there was a consensus at hand. But this is the list of different consensi. And so clearly there's a sense that we're onto something, but also there seems to be a wide variety of things that we're onto. And so I think maybe one way forward is to think about the stories of particular groups and their patterns, the histories, if you will. And so work like the work that Phil Ward has so greatly done over the years. One thing maybe worth noting, though, is that one of the hypotheses that was really prominent in people's responses, but is not prominent in literature, is the idea that age is really important, the age of biomes. Older idea that essentially faded for a while, but it's really hard to test. So I think there's some interesting things there. And so we're trying to work at seeing in other ways, too. And so this is a way of seeing scientific community. What do we actually think? This is remarkably hard to get a sense of. But we're now moving toward looking at the particular group, though, to look at the history of the ants per se and their diversity patterns. And Andrea Lucky, who was here at Davis, is now working with us in the lab to try to get at some of these questions of that history. There's Andrea. Andrea informs me that in Papua New Guinea, this is a very funny picture because women never wear flowers. And so far, our reconstructions suggest some of what's already in the literature and some new things. That Andrea's found ants ancestrally reconstructed as tropical, mostly suspected that, but with recent shifts into colder realms, those places that are less diverse. We've also shown separately that the traits to deal with the extreme hot and modest cold are difficult to evolve. And the diversity patterns may be a function of this and other histories, that time component. That's very much early in progress. And what we really want to get at now is to the issue of consequences in biomass and just how much ants. You often hear this quote that 40% of ants in the 40% of biomass in the tropics of animal biomass is ants. That comes from a study where they shot all the mammals and birds they could, and then they asked a friend how many insects there were, literally. So we really don't know, but we're starting to try to do this. And so we've moved to a next stage of this global collaboration, where we're now up to 87 collaborators globally and keep adding. And we're also enlisting children to do our version of a breeding bird survey, but with kids around the world sampling ants, and we're targeting cities and then places we know that there are lots of species still to be discovered, to get kids out in their backyards to document what's there and what we're missing. And we've already had some new state records turn up from doing this in North Carolina. So it's a pretty exciting endeavor to get at the process. And so we're doing a lot of that, bringing people together to see what none of us see on our own. But we're also doing more simply is to try to see the future by looking around, by looking at what are the stories of species in the environments really directly around us. So I think it's really interesting to be on campus here and to think about ants. And the story is that here there are very few native ants because all of the ants on campus are Argentine ants. But it would be interesting to see whether that's really true if you did some really intensive sampling, whether you can turn up some things high up in trees and what's going on. And it just takes looking locally. And in particular, I've been really fascinated with thinking about this in the context of cities. If cities are part of what the future is, if we just look at the cities around us, what do we see? And I think the main story we tend to tell ourselves about cities is that they're places where we get trashy species like pigeons. But the crazy thing about pigeons is that they're rock doves. And they're not trashy species. They're not generalists. They have a very specific story and a very specific life history. They're cliff nesters. Do well in cities because we reproduce things like cliffs. And so we've started to take this approach of thinking about urban species and their particular life history traits and thinking about how they are doing well in those places that we're expanding. And just an indication that these cliff-like environments are often very cliff-like, where we've spread rock doves. But the difficulty of seeing these places right around us is again, really literally in seeing. The breeding bird surveys, our best measure of what birds are doing, they omit cities. And so if a bird route you've surveyed over and over again becomes urbanized, you cut it out. If you look at satellite data of cities, if you look at, so, for example, MODIS data of productivity, primary production in cities, They cut the city out and then replaced the city with the productivity that would be there were the city not there. And so in a lot of really specific ways, we really are especially blind to the species right around us, and that includes really abundant species like rock doves, that because they're abundant, we assume somebody knows about them. I love bullfrogs in this context, or, the Paramiscus mice. Bullfrogs, it was only 5 or 10 years ago that it was realized project sounds, low sounds out their ears. Bullfrogs are potentially one of the best studied organisms on Earth, yet we're missing basic things. And we miss it in part, I think, because we assume the things that are around us are well understood. And so we've been spending a lot of time thinking about the things really right at hand. And Sean Menke, who was a postdoc at my lab now at Lake Forest, did the very simple thing of just going out in Raleigh, North Carolina, and doing a structured sample across Raleigh to see who's there, which went really smoothly, except that he accidentally ended up in the front yard of my other student, Naima Harris, who was not happy to be greeted at 7 o'clock in the morning with ant sampling by Sean, who seemed like he was being creepy rather than randomly sampling her yard. But aside from that, it went all well. So Sean just wanted to see who was there. And so what we end up with is that Raleigh is a very diverse city. So we get about 100 species of ants in Raleigh, depending on how we define the city, greater than 70 species on campus at the university, which I think is pretty astounding. And that includes new records for the state, potentially new species, a strange rare species, a cryptic invader, and a mutant species. I'll talk about a couple of these. And a lot of this work with Sean was done by undergrads, Adriana Cabezas. We did a lot of basic stuff just to see who's around. One of the strangest species is what I took to calling the tiger ant. And I admit that doing this only because this was a 17-year-old student who came to work in the lab and said she only wanted to work on tigers. And we didn't have them, and this ant didn't have a common name. And so she found on campus an incredibly rare ant that's been sampled only a few times in the U.S., Scothria testacea. And she, along with Andrea Lucky, was able to get it out of samples alive, and so saw this ant for the first time alive anywhere in the world on campus 50 feet from where we teach intrabio. And it just took looking. And so here Catherine Driscoll is presenting this work. Not so long ago, Benoit Guénard in my group was taking pictures right around the city in this sort of Cary, this suburban city next to Raleigh, and he found this ant, which he took pictures of and put in the web. It turns out that this ant is what we've been calling a Bigfoot ant, that it was described 60 years ago on the basis of one specimen. Many people thought it was fraudulent, that it wasn't a good species. It was never seen again. Benoit saw it right next to his house, right next to the city. That's the good news. The bad news is he took pictures of it but didn't get any specimens, and so this is still all we know about it. But, I mean, this ant is within a stone's throw of 700,000 people. And this is all we know about it. Fun. Benoit has also worked a lot on this ant, which he turned up early in his PhD in Raleigh, North Carolina, and turns out to be the most common ant in the county we live in, Wake County, and was introduced to the U.S. in the 1930s, 1920s, and sort of cryptically, without anybody paying much attention to it, became incredibly common. And so Benoit has just focused on this ant around Raleigh, and he's since gone to urban Japan, shown that it actually has a way more widespread distribution than anybody knew. Documented the invasion. In the process of documenting the invasion, accidentally turned up a new species of this genus in Japan. Observed a totally new kind of foraging of this ant in its invaded range. It's only the 7th kind of foraging documented in ants. They actually carry other workers to food, show them where it is. That's their recruitment strategy. And documented this ant has widespread consequences and modest panic. The panic comes from this. It has a 2 in 100 anaphylactic shock rate, which is twice that of honeybees. So it's pretty bad news. Mostly it's pretty peaceful, though, which is good. And this was really just by looking. Benoit was looking for something else, found this ant, and we kept exploring it. And then Sean, in doing his urban work, the mutant ant came up. And so this mutant ant is Tapinoma seseli, and the pest control agents in Raleigh as elsewhere think that this is a crazy ant doing weird things. I said, 20 years ago, I don't believe we had nearly the calls about odorous house ants. We would have still had a large number of calls about ants, but they were different kinds of ants. Our number one pest. And so here's this ant that everybody thinks is spreading across cities. It's some new mutant form. It's moving through the cities, East Coast, West Coast, up into Colorado. So Sean just started looking at it and it's doing some really basic work. He's able to figure out that the urban forms do seem to have many queens. They have large colonies, little aggression, different body forms. So it lends credence to this idea of the mutant. But then he actually just looked at some basic genetic structure in this ant and found that there are probably a group of different species of what we're calling Tapinoma seseli, and they've moved into cities independently. So here's this mutant form is actually this amazing life history transition that consistently occurs in cities. But again and again, they're moving into cities and doing the same thing. And so really just by looking. So from no fewer than 4 clades, each time take on a large colony many-queen form. The natural colonies are more traditional, but less polygamous. And urban colonies can span blocks. But the other question that came up just in looking around us were how these ants were distributed in the city. Because the story we tend to tell is that the more disturbed habitats have fewer species and they're bad. And then we focus on the less disturbed places. Well, Sean started to focus on all these different kinds of habitats and look at how they were different. And what he found was, I'm not showing it in this slide, but that these different habitats tended to have similar diversity. But then when he started to look at where these species were found, he found something very different. But if you look in the forest up here, you have species with a minimum latitude that's relatively far north and a minimum longitude relatively far east. But the further you get into really intensive urban environments, you get species that are essentially shifting up from the southwest, dry adapted species. And we've now done thermal tests on these species, and we can show that the species you're finding in these habitats are the species with the greatest thermal tolerance. And so in cities, we're favoring this subset of species that are not trashy. They have specific life history traits that are different and interesting. And this all becomes really interesting as we think about whole cities and how they're changing and how these species are moving around them. This is a model of the heat island effect in Atlanta, where some parts of the city are 15 degrees C above other parts on average mean annual. So a big effect. And so we think that maybe some of these species are moving north into unfamiliar territory. Maybe this is evidence that other species are also finding themselves in places they're not familiar. I don't know if it's real, but it's too funny not to include it. And we've moved to starting to look at this in Raleigh, and so we're actually starting to sample in Raleigh really hot places and really cold places. This is Raleigh here, and the red places are 10 degrees C hotter than the blue places, mean annual temperature. This is the RDU International Airport. This is Umstead Forest. So we have trees right here that are experiencing 10 degrees cooler conditions than right here. And so really interesting things could be going on there. We're super excited to start to. And now quickly I'm going to talk to you about one more way that we're trying to see the future. And that's with experiments. And so as we all know, one aspect of the future is that it'll be warmer, both because of global change, but also because of urbanization. Even before we get any global climate change, we're going to get these heat island effects, which are huge. We need ways to think about this. I think there's a fair argument that ants are interesting climate lab rats in as much as they respond socially in the way that we respond socially. So maybe there's some ways to think about them, about rats for physiological processes. This is an old idea that we can think about them like us. This is a Kaiapo wasp nest. And these are the sky layers, the earth layers, with the different wasps occupying different parts of this social strata. And in effect, thinking about the wasps like they think about themselves. In some ways, we know a lot about what we might predict in the future from all the other things we've done. So based on the global ant data, we can say that maybe we'll expect more ant genera regionally if we just substitute space for time. Might expect more ant species. Locally, greater ant abundance and biomass, more canopy ant species if precipitation's high. But all this is dripping with assumptions. People do it all the time. But it's really hard to know if using space as a substitute for time tells us a lot about the future. We need other ways to see. We need other ways to pull back that ocean. And another way to do this is we can model individual species And so these are models for all the ant genera of North America. And these are areas with lots of turnover. So with full dispersal, we get lots of turnover here in these predictions. With less dispersal, we still get it in the same sorts of places, but slightly different patterns. And there are lots and lots of papers like this, modeling individual species or groups of species, but it's hard to know whether to believe them. And we can use other approaches. We can actually look at the thermal tolerance of species and see if that would predict where they're going to go. And so Sarah Diamond in my lab has measured thermal tolerance in 240 ant species globally to look at how that varies geographically in response to other things. And one of the things we find is that the distribution of species and their thermal tolerance don't match in just a simple way, which is one of the things we assume if we run species distribution models. And so there's a problem. And we're starting to try to reconstruct thermal tolerance in the phylogeny. So this is the maximum temperature species survive at. And so you can see one of the things to see is that there are whole clades. These are cold, not hot tolerant species with negative numbers. There are whole clades that don't take much temperature. They're not good at hot temperatures. And other clades that seem mostly really good at hot temperatures. So there's some phylogenetic signal here. But there's also a really strong geographic signal that's super interesting. We're just starting to explore. This is the warming tolerance of species. It's their maximum tolerance relative to the maximum temperature they experience. And so we found that tropical species, even if they have a high maximum tolerance, it's pretty close to the temperature they actually experience. If you go out of the tropics, you get these species with much greater tolerance than they actually experience. So there are really interesting things going on here that might not match with simple assumptions. And so what we need to do is figure out some other way to figure out what really happens. What do we see on the ground? We started off by making these small chambers where we could warm the leaf litter about a ½ degree to a degree to look at local responses. And so we see ants responding locally the way we would predict. We did this in Harvard Forest in Massachusetts and Duke Forest, North Carolina. And they look similar enough from this angle that I can't actually remember which one this is, Harvard Forest. And so we looked at all kinds of things the ants do: predation, granivory, nectar removal, seed dispersal. And I'll just give you the take-homes, which is that so far we see less seed dispersal, less nectar removal, less grain removal, and somewhat more predation with just a degree of warming. And the most common native species becomes more rare, and this only happens in North Carolina and Harvard Forest, where we expect the biggest change. We see nothing. And so we felt like we needed to go bigger. I forgot to mention that one of the species that becomes really common, the most abundant by far, is Corenogaster lineata. Funny. Fortunately, we know very little about this ant, but suddenly, with just a little bit of warming, it's very, very common. But we've decided to move on from this with this motley group. So I'm in the middle. It's Nick Catelli, Nate Sanders, and Aaron Ellison. And so I led a grant to expand that project. That's not actually it. This is it, but to make bigger chambers, to repeat the same kinds of experiments. And so this was a crazy engineering feat, and I felt like a general contractor for big parts of the last couple of years. This is just for one chamber. But this is what they look like. So we have these chambers out in the forest now. They're open top that we can warm up to 5.5 degrees C above ambient and look at responses not just of ants, but of lots of other things too. These are plena that heated air flows through, and it sort of trickles out into these chambers. You can see oak seedlings regenerating in here, and each chamber is around one of these trees, and we have 12 of these at Harvard Forest, 12 at Duke Forest, and these are paired with plots that Jerry Melillo and Jim Clark have for plants. So in total, we have something like 48 chambers. So there's oops. That's my 5 a.m. coming in, sorry. This is just looking into one of the chambers. We try to access them through these windows to disturb them as little as possible. And so these have only been on for a year, but we're already seeing interesting things. One of them is that this same species is becoming super common. Seed dispersing ants are becoming rare. Seed dispersal rates are slowing down. Plant herbivory rates are increasing. And the plant phenologies are shifting dramatically, almost a full month. But so far, the ant phenologies, when the ants start foraging in the year, haven't responded. The ants are responding, though, day-night. So we see a shift in night. And so we have lots more to do here, but we're trying to use these chambers to see what we can't see otherwise. This is just a visual representation of some of these changes. This is on the same day, and you can see that these 5-degree chambers have greened up much more at this same point. These controls are actually still quite brown. It's the same day of the year, same site. And so what we want to move to is from this to try to predict who is favored and see if that's actually what happens. And so we'll get there soon, but we're not quite there yet. What we have so far is to be able to look at the maximum temperature recorded in the species range and its maximum thermal tolerance. And so there's not a good relationship. So if you experience really warm conditions, you don't necessarily experience you're not necessarily any more predisposed to dealing with the heat. But what these red circles are species that are doing really well in our chambers. And then these squares are the species that are doing the best. Blue and black are species that are actually declining in abundance. And so it seems as though we might be seeing a signal already of thermal tolerance, but not distributional sorts of effects. And so again, we're trying to integrate these ways of seeing, and so now we're trying to bring lots of people into seeing these chambers, just in the way we brought lots of people to seeing the whole world. So we're bringing people with different expertise in to look at different groups. So springtails, scale insects, decomposition, herbivory, ambrosia beetles, and other groups. And there's lots of room for people if anybody's interested in being involved. Because essentially we've created these miniature worlds, and there are so many things we can look at to try to capture what's happening in them. We just did a big BioBlitz chamber sampling at night with school children. So we brought students and scientists out at night to sample these with black lights, covered them in black lights, to get a really broad picture of what's going on. So we've been having a lot of fun. And so we're trying to bring this all back together to think about history. And I have to admit, there's a little bias here today that I've started by talking about small things in general, but I finished talking mostly about ants. But the other thing that's worth remembering is that in the story of this book, the story of revealing the sea, doesn't quite have a happy ending. That the boy who holds back the ocean can't hold it back forever, and the little boy drowns. And so by focusing on ants, I'm trying to keep from drowning. It's a big world of unknown, and we have to start somewhere. But it really is a wonderful unknown world until which lots of us sail our ships. And I have a lot of fun doing it. And I do it with the help of all these people back in the lab pulling the oars I mentioned today. So thanks to all these folks, including Sarah Darlampro, who's in the back. Sarah, where are you there? This was in here before you, yeah, I was even coming out here. But thanks to you for coming today, for putting up with my sleep deprivation a little bit here. Questions? Down to 5 centimeters, pretty good. If we get down to 10 centimeters, it trails off. But clearly different than the predictions of what soil temp will do in general. So our chambers just have air heating, which permeates down into the soil. Jim Clark and Dre Malila's chambers have soil cables. So their soil is a little bit hotter, a little bit deeper. And so we'll be able to compare what those two look like. The thing about soil cables and ants is that at least fire ants are attracted to soil cables, and so we avoided them. But we do have that comparison. Particular context for that question? Just interested in the soil?
Studying ant control in context of agriculture. So, and I've seen some pretty heat tolerant ants out in Coachella Valley. Pretty amazing.
So in our site, Chromatogaster and then a suite of Monomorium species are about 7 degrees more tolerant than any of the other species we have out there. And they do seem to be doing much better.
Probably 120 degrees soil surface.
Yeah.
Have no problem.
So if you look at the phylogeny of ants, there are the super heat tolerance. It does turn up more rarely. Like modest heat tolerance is sort of more labile. But tolerating above 50 C, it turns up in relatively few clades that are pretty good. Yeah. So one of the ones that's like that is Monomorium as well. And all of our Monomorium species, even from very cold environments, are very heat tolerant. And interestingly, they've also really succeeded globally in urban environments. Interesting to think.
Mechanisms that they're based.
Yeah, I doubt it's the same for the ones we know about. It's not always the same mechanistically. Question back?
So I was curious if heat tolerance, are there any correlates of that kind of metabolically or functionally in ants, or is it really idiosyncratic? Is it decoupled from the rest?
We don't know metabolically yet. It's knowable, but like you could look at heat shock proteins would be one thing to look at. We've not done it, but it's certainly possible.
Their limit. They're killing
Yeah, so it may be a performance curve sort of issue that if you shift out the whole performance curve because the max goes out, that they're also just doing better at those higher temperatures. So it's not, in that case, the critical maximum would be a proxy for this whole thermal performance curve shifting. We've started to look at tolerance of CT min. It's more difficult, but it'll be interesting to see if they shift together. If you evolve more thermal tolerance, do you lose tolerance of cold conditions? There are reasons to think both ways.
Do you know what they're optimizing? Say that they're more tolerant, presumably that means that they are resistant to dying at the higher temperatures. But if the ones that are more tolerant, they actually choose higher temperatures to go to, and if so, what are they optimizing when they do that?
So we've looked at 2 species so far from our chamber sites where we've done running speeds. So this is Heidi McLean, a student of Lauren Buckley at UNC. So she's done running speeds and actual thermal performance curves for 2 species. They turn out not to be really important species in our story. So ask me again in the future, but I'm very interested to know what.
Just say an observation. If you look at higher altitude grasshoppers, which grow up at cooler temperatures, pick hotter temperatures than the lower altitude grasshoppers do, and what they're optimizing is reproduction.
So
They've got a very short season up there, and they'll find as hot a place as they can.
that's interesting. So one window we have into that, we haven't analyzed the data yet, but we did a common garden experiment where we brought ants from 6 species from Florida, North Carolina, Harvard Forest, all into a phytotron, an indoor facility, where we manipulated temperatures to coincide with summer temperatures of those 3 sites. And the ants from the cold conditions were much more active at the hottest temperatures and seemed, well, we haven't analyzed the data yet, but there's some interesting things going on there in terms of what their behavior is relative to their performance. They don't seem to match up perfectly, which may have something to do with that sort of phenomena. That's really interesting. There's a question over here I missed.
Yeah, the thing in your chambers, measuring predation and seed dispersal and things like that, and attributing what you're seeing to the change in temperature. So I'm just wondering, it is a protected environment, right? So is there a way for you to factor out other? It's an undisturbed environment, for example, and is there a way to deal with that you considered, or?
In what sense? Could you elaborate a little more?
Well, I'm thinking that you've got this. You're creating these chambers, basically. And so you have ant species in there, you're manipulating temperature, but compared to what would be the absence of the chamber, you'd have a lot more seeds dispersing into the area. It would be, it's not a natural environment. It's kind of an artificial chamber where you're, and I realize you have to start somewhere, but I'm just thinking, is there a way to maybe factor out factor in some of the differences between what you're doing and a natural setting?
Yeah, so that's a very good point. So that like any of these ways of seeing, the chambers come with biases, and so one of them is what's often called a chamber effect. That the chambers have a treatment effect, but also by dint of being a chamber, create different conditions. We've tried to get at that multiple ways, and so most of the experiments we do in the chambers, we do in no chamber controls. Also more broadly in the landscape, we're trying to do these lab manipulations where things are totally in our control. But yeah, one of the interesting places for sure we see the chamber effect is we were really interested in ticks initially, tick responses, which was totally naive, it turned out, because most of the ticks in our environment are deer ticks. We do see ticks totally disappear in our chambers, right? Because the deer don't, with one bad day exception, don't tend to get in. Yeah. We had a small deer in one of the chambers at one point and did not want out. Copperheads also like them. Louis?
I don't know if I've noticed this. You mentioned earlier, but how porous are the chambers, the ant movement in and out?
It differs again from our chambers relative to Jim and Jerry's, but our chambers are totally porous, so they can move out. So I think of them as really best replicating like what's happening in urban forests as urban forests warm. So one of the first responses the ants can do is to behaviorally respond to conditions, maybe in some ways related to some of the Baldwin things your students thinking about. But and so they're open topped, and so things can move in and out. We've had big discussions about for the next round whether to close them off totally. And we don't have a consensus yet. There are advantages and disadvantages to both. Yeah.
Thermal tolerance, and I wanted a bit more information about how the temperature has changed. So you're talking about you change it by 5 degrees. Is it a sort of press 5 degrees across all seasons, or
So it's a press across all seasons. And
so it's just starting. That's your starting.
Yeah, and so Judith Canner, who is a PhD student with Kevin Gross and myself, and is now at CSU Monterey Bay, she did a simulation where she simulated our warming experiment, and she did it as a press, and then she repeated it, but allowed it to occur over 100 years, and looked at responses of ants based on what we already know and responses of plant demographics. And so up to 4 degrees in those simulations, which come with their own set of assumptions, the press and the long-term experiment were very similar. But once you got to 5 degrees, they diverged. And so that's another interesting thing. Although that makes it more like, I think, what happens in cities. Because as you build a city is a press experiment.
Yeah, does it end up, right, so that makes sense then. 'Cause I'm thinking about, like, we think about natural systems where, like, some of what Louis is playing around with snowpack and things, it's not only, may get changes in temperature that are, with respect to the soil and the surface fauna, that behave in very different than consistent throughout the year. They're just sort of short times where there's spikes in certain seasons, but not on others. So your decision to do that, what was the thinking? Is that?
Well, so I mean, one of the issues with these is you only have so much control over temperature, and so you want to maximize your ability to institute the treatment you want. And so the chambers people have used before have not gotten as hot as these, and so that was really a focus to try to get these a little closer to 2100, 2050 even models, which are hot. One of the things we can do to get a little bit more like at a pulse sort of phenomenon is we take ant colonies, we put them in just to overwinter. And so we can look at the response to having a winter interval of warming on specific species. But we looked at, so the Occam chambers at Oak Ridge, I don't know if people know those, but they're pretty famous chambers for warming in plants. But they actually have about a degree and a half of wiggle on every treatment. And so when we were starting to build these and looked at that, it gave us concern about trying to do anything really complicated in terms of spikes in temperature, things like that. But we have the ability, we could do it in the future, but it's going to have a lot of error. Yeah, any question in the back?
Sampling in cities, and kind of, did you surveyed in parks and around Raleigh and around other parks where these samples were done? Did you look at any agricultural systems like vineyards?
So in Sean Menke's work in Raleigh, it was a stratified random sample. So he included agricultural fields in the city, which didn't get any vineyards. Got some tobacco, some wheat, things like that, but not tons of samples of any individual type. But my intuition is that if we do more work in some of those, especially tobacco and cotton, that we ought to be favoring some really thermally tolerant things there. Those are nasty, difficult living places. But I'm really fascinated by at least from the literature that I come from, the perspective on those landscapes is often a conservation perspective. Well, we lose all these species. We're left with a couple. But I think thinking about what those couple are and how they're dealing with those conditions, I think it's fascinating because there's the incredible environments we're spreading everywhere. So whoever is able to deal with those conditions, they're going to be around us. So at least the sort of know thy neighbor perspective. It's
Jersey things. I haven't identified the ants as of yet, but it looks several dozen species.
That's interesting. I'd love to see those at some point. We did Broadway in Manhattan, and that was a fascinating one. And we're going to revisit that with some more sampling. All kinds of unique Manhattan problems, like people pooping in pitfall traps. But you do what you've got to, yeah. That attracted a different fauna, incidentally. Question in the back?
I was wondering if you could talk a little bit more about what kinds of methods.
So we're sort of doing battle with that right now. So we're using a mix of techniques, and so we're going to we have a bait kit that we're going to send out to schools anywhere that people are willing to do it. And because of the global collaborators, we can get these people to get into schools all around the world. And we know that there are places like Myanmar where there are 20 ant genera known, and they're based on its climate, we'd predict like 50 ant genera. So places like that, a school kid with a couple of vials is going to find new stuff. We're trying to target some of those places. But it's also become apparent in starting to do this that there's a lot of advantage to doing things locally enough that we can go into the schools ourselves, revisit schools and work with teachers. And Bonnie's going to be a part of an event this spring where we're going to try to get some teachers up into trees around Raleigh to sample in the trees, which I think will be really neat. And the students will be there watching. And there'll be some, neat species that we don't see very often. One of these Camponotus that has one of the Camponotus ant species that has a really flat head that blocks off its nest entrance, we never collect, but it falls on us sometimes in the forest. So we know it's up there. And I think it'd be pretty easy to find it in some of these school playgrounds. So to find this rare ant species, have the kids see that. I think that's really exciting. I've really come to enjoy working with the students doing that. Teachers are more complicated, but the students are great. Did that answer your question? I'm glad to talk more about it later, but yeah. Other questions? Well, thank you so much, y'all. I really enjoyed my visit so far.